A cyclic diterpenoid has neuroprotective effect against Gulf War illness involved neurotoxicants. This is presented by Felixe Rivera Moctezuma, undergrad voluntary student of Dr. Sabeba's and Dr. Frischmann's laboratory from the Neuroscience department in collaboration with the Biochemistry department at Universidad Central del Caribe. Organophosphate compounds have been widely used as agricultural and household pesticides, jet engine lubricants, and warfare nerve agents. Therefore, the general population and military personnel could be exposed to organophosphates, not only during combat or terroristic attacks, but also during routine military, industrial, or private activities. Delayed neuronal damage of organophosphate intoxication it is thought to contribute to neuronal death and various neurological illnesses, such as the Gulf War illness. Gulf War illness is currently an untreatable multi-symptom disorder experienced by more than 250,000 veterans from the Persian Gulf War between 1990 and 1991. The distinctive hallmark of the Gulf War illness includes chronic fatigue, migraine, muscle and joint pain, gastrointestinal problems, and cognitive disturbances such as depression and anxiety. Various studies have consistently linked these symptoms to exposure to pyridostigmine, DEET, permethrin, and traces of sarin, the most commonly used nerve agent. Currently, there is no effective cure for the Gulf War illness or the chronic effect of other neurotoxicants, but we might have the solution. 4R is a cyclic diterpenoid with anti-inflammatory and anti-apoptotic properties. In ex vivo hippocampal slices, 4R protects against NMDA-induced excitotoxicity, reduces oxygen-glucose deprivation damage, and neurotoxic organophosphates such as pyroxene and a surrogate of sarin, the isopropyl fluorophosphate, or also known as DFP. Administration of 4R to rats 1 hour prior or 24 hours after treatment with DFP dramatically reduces neuronal death and inflammation in the hippocampus. 4R crosses the blood-brain barrier and it reaches higher concentration in the brain than in plasma, regardless of the administration route. Therefore, we hypothesize that the loss of neuronal function due to DFP can be reversed by the application of 4R. To investigate the neuroprotective potential of 4R, we employed our well-proven and published method using ex vivo rat hippocampal slices. We tested the hippocampal slices' ability to produce synaptically evoked action potential, measured as population spikes, in a population of about 60 neurons. Thus, the size of the population spikes is proportional to the number of electrophysically active neurons in a slice. In our preliminary experiments, the hippocampal slices were distributed in the incubation chamber among 3 lanes, as demonstrated on the picture. 1 hour after dissection, the Schaefer incoming axons of each slide were stimulated with a stimulus double the strength required to elicit a threshold population spike. The initial response was recorded as a population spike area. Lanes 2 and 3 were exposed to the neurodegenerative agent for 10 minutes and then watched for 60 minutes. After this period of time, lane 3 was treated with 4R for 60 minutes. A final populational spike was recorded, elicited by the same stimulus recorded from the same position as the initial response. To compare them after the experimental treatment was finished. Here we show 4R percentage of recovery population spikes, where the control is at 100% and DFP reduces the recovery percentage down to 40%. Both pyridostigmine and DEET reduced the recovery to 50% and permethrin to 60%. The presence of 4R shows recovery of population spikes, suggesting that 4R has neuroprotective effects. After histological ultrastructural analysis of hippocampal slices employing transmission electron microscopy, we observed that DFP causes swelling of the mitochondria and loss of the membrane CRISTAE, which was then restored after the application of 4R. To elucidate the signaling mechanism triggered by the application of 4R, we targeted a well-known molecule involved in pro-survival signaling pathways, AKT protein kinase B. We performed a timeline experiment in the presence of 4R alone, and we observed a decrease in phosphorylation until 20 minutes. Then, at 30 minutes, increased to 125%, reaching a plateau between 30 and 60 minutes. However, in the presence of DFP, we see that the decrease in phosphorylation was more significant at 20 minutes and increased to 100% in 60 minutes. To summarize what we have discussed, 4R sembranoid has strong neuroprotective effect against Gulf War agents, ameliorates ultrastructural changes in hippocampal neurons, and also could reverse the organophosphate neurotoxic and neurodegenerative effects. In conclusion, our study suggests that 4R activates AKT PKB pro-survival signaling pathways. We want to thank NIH for sponsoring these experiments.